You know the look. We've all seen it, right? That kind of sad-looking houseplant or those weird brown spots on the leaves in the garden? And when we see a sick plant, what do? We give it some water, maybe a little fertilizer, and just hope for the best. But what if the real problem is something you can't even see? What's actually going on down at the microscopic level?
Sometimes, the problem is so much more complicated than just a lack of water or, a hungry caterpillar. I mean, look at this. This isn't damage from something. It's a structure that was created by an invisible hijacker. Something has literally taken over the plant's biology and forced it to grow this bizarre, almost alien-looking thing.
Okay, so it's one thing if a single plant in your house gets sick. It's a bummer, but that's it. But what happens when this kind of thing spreads, not just across a field, but across an entire region, a whole country? Well, the consequences can be absolutely devastating.
2 million. Just let that number sink in for a second. That's not a measure of lost crops. That's the estimated number of people who died during the Great Bengal Famine of 1943. And the cause? It wasn't a war. It wasn't a drought. It was a fungus. A single type of fungus that caused little brown spots on rice plants, and it led to a catastrophic famine. This shows you understanding plant disease isn't just a fun gardening hobby. It is essential for our survival.
This is where the heroes of our story show up. See, there's an entire field of science that's dedicated to preventing these kinds of disasters. It's a group of scientists who basically act like crime scene investigators for the plant world. They're the detectives defending our food supply.
The official name for it is plant pathology, or if you want to sound really smart, phytopathology. And that word is actually pretty cool when you break it down. Phyto is Greek for plant, pathos means disease or suffering, and logos means knowledge. So it's literally the knowledge of plant suffering. Their entire job is to understand and protect the plants that feed all of us. So what does a plant detective's job description look like? First, they have to figure out the cause. Is it a living thing, like a fungus, something non-living, like a mineral deficiency, or is it something in the environment, like a drought? Then, they try to understand the disease's whole life cycle, analyzing the battle between the plant's immune system and the attacker. And finally, with all that intel, they figure out how to manage it and hopefully stop it from happening again.
Okay, so just like any good detective, the very first step is to get to the crime scene and start looking for clues. For a plant pathologist, the plant itself is the crime scene, and the first clues they're looking for are those visible signs of trouble.
Now, these detectives make a really critical distinction between a symptom and a sign. A symptom is just the plant's reaction. It's wilting or its leaves are turning yellow. It's the plant screaming, "Hey, something is wrong," but it doesn't tell you what's wrong. A sign, on the other hand, is the smoking gun. It's the physical evidence of the bad guy, like you can actually see the mold growing or you find the insect larvae. Finding a sign is like finding fingerprints at the scene of the crime.
Let's take a look at some of these clues. What we're seeing here is a classic symptom called blotch. Notice how these aren't neat little circles? They're big, messy, irregular shapes of dead tissue. That kind of spreading lesion is a huge red flag that a fungal or bacterial infection is taking hold.
Different diseases also leave behind their own unique visual signatures. So on the left, you see anthracnose. See how it has those sunken, target-like lesions? That's really focused damage. But then on the right, you've got blight. That is just a wave of destruction. It's super aggressive, super rapid, and it can wipe out huge parts of the plant. Two totally different patterns of attack.
And then you get something really dramatic, like this gall. This is basically a plant tumor, this huge chaotic swelling of tissue. It happens when a pathogen or an insect hijacks the plant's own growth systems and forces it to build a custom home for the invader. It's just wild. So a visual inspection is a powerful first step, no doubt. But to really, truly ID the suspect, our detectives have to take the evidence back to the lab. Why? Because sometimes the most important clues are completely invisible.
And welcome to the forensics lab of plant pathology. This is where the high-tech tools come out to play. This is where scientists can look right past the symptoms and identify the precise genetic fingerprint of the microscopic culprit that's causing all the trouble.
And this quote from a textbook on the subject just says it all. The absolute best, most accurate diagnosis you can get comes from combining two things: looking at the morphologic changes, which is just the sciencey term for the visual symptoms we just looked at, and molecular testing, which is the DNA work they do in the lab. You need both. You need the evidence from the crime scene and the analysis from the lab.
So, what's their number one forensic tool? It's a technology you've probably heard of, PCR.
The absolute best way to think about PCR, or polymerase chain reaction, is as a genetic photocopier. It can find one single tiny piece of a pathogen's DNA in a plant sample and turn it into millions, or even billions, of copies in just a couple of hours.
So how does this genetic photocopier actually work? Well, it's a repeating cycle. First, you heat the DNA to unzip it into two separate strands. Then you add these custom-built little things called primers that are designed to find and latch onto a DNA sequence that's totally unique to your suspect. Then, a special enzyme comes along and copies that section. You just repeat that cycle 30 or 40 times, and in a few hours, you've got billions of copies, more than enough to make a positive ID.
Now, not all PCR is created equal. The original method was, well, it was a bit slow and it needed a lot of extra steps. Today, the gold standard is real-time PCR. It uses fluorescence to literally watch the copying as it happens. This makes it faster, much safer from contamination, and in the end, it's actually cheaper. It's a massive upgrade for our detectives. And PCR is just one tool in a huge and growing toolkit. Scientists can also use things like DNA fingerprinting to identify very specific strains of a pathogen. They have tests called ELISA that detect unique proteins instead of DNA. And they have molecular markers that act like little genetic flags to pinpoint a known sequence on a chromosome.
So we've gone all the way from a sick leaf to a high-tech forensics lab. Which brings us to the big picture. Where is all of this heading? How is this science going to protect our food supply in the decades to come?
And the future? Well, the future is even more advanced. Just imagine a single microchip that can test a plant sample, not just for one disease, but for thousands of known pathogens all at once. It would be like running a background check on every possible suspect simultaneously. This would let us make faster, more complete diagnoses and help us catch outbreaks before they even get started. From the Irish potato famine to the Bengal famine, history has shown us time and again the devastating power of plant disease. Today, these scientific detectives are on the front lines, using visual clues and molecular forensics to defend our fields. So, the final question to think about is this. In a world with a changing climate and with global trade that can spread new pathogens in an instant, how will these plant detectives continue to innovate and protect our future?